Hi students, in this video I am going to discuss about the proteins, polysaccharides and nucleic acids which are in the biomolecules. It is more important for the NEET exam because so many types of the assertion reason and statements and multiple choice questions can be arised. Already he asked 5 questions in all the 3 subheadings. What are they? In the sequence I will discuss those things. Now what is the common between all the three? All they are the polymers. Polymers are the made up of monomers. And they are the big molecules. That are macro biomolecules. And in this one, proteins and all these three macromolecules where they are present already we studied, that is in the acid non-soluble pool retentate it is present. And in this one more common point he will ask in the NEET. What is the common point? What is they are the made up of monomers. They are present in the retentate. They are non-acid soluble. They are not soluble in the acid. So these are the common points. And now whenever we are seeing proteins are made up of the monomeric compound, monomers of amino acids. Polysaccharides are made up of monomers of the sugars. And next, nucleic acids are the made up of monomers of the nucleotides. These are the common things which are important for the assertion reason statement also. Now one by one we will see. Now first one is the proteins. What is protein? Protein is basic body building units of the cell. Cell will be built up by the protein and enzymes are the proteins. So enzymes are most important. These proteins are most important one. So proteins are the polypeptides, linear chain of amino acids with peptide bonds. What is this? It is linear. You see, amino acids are present in the line, and because it is polymer made up of the monomers. And next is it is amino acid with peptide bonds. Between the two amino acids, what the bond is to present, that is called a peptide bond. So they are called polypeptides. They are also called as a polypeptides. So proteins are the polypeptides, and they are arranged in the linear, and with what the bonds polypeptide, why we are calling peptide bonds are present in between them. Now whenever we are seeing how these things, again polymers are the two types, homopolymers, heteropolymers. And in this one monomers, all the monomers are the only one type of monomers are present in the polymer, that is called as homopolymer. Different types of the monomers are present in the formation of the polymer, then it is called as a heteropolymer. The protein is heteropolymer, means no protein is present with the same type of the amino acids. But why we are telling, and why he is telling in the textbook also, out of all the three, only homopolymers are formed only in polysaccharides. Remember, it is most important. And nucleic acids, proteins, they won't form the homopolymers. They are the heteropolymers. Remember it. And next is heteropolymers with 20 types. In this heteropolymer, it is monomers. Monomers are so many kinds of the monomers are there. There are 20 amino acids, 20 types of the amino acids used to participate in the protein formation. So these 20 types of the protein means the 20 types of amino acids. Nearly 100 amino acids are there, but only 20 will participate in the formation of the proteins. So, monomer number in the protein formation is number of monomers which can participate in the protein means that is asking 20 amino acids. So, here and now whenever we are seeing here, one more thing, this homopolymer, heteropolymer, this is the all the proteins are the heteropolymers only, no homopolymer can be seen. And next, whenever seeing the amino acids, these are the amino acids which are present. And now you know that in the digestive human digestive system from the 8th class onwards you are studying. Different enzymes used to present in the digestive system. Protease used to convert proteins into the amino acids. You have studied or not? That protease, what it will do? It will break this peptide bonds. And then the amino acids will be used by our body to form our wanted protein. Our proteins are different proteins. How it will be? Amino acids will be supplied from the plants only. Plants only will synthesize all type of the amino acids. But animals cannot synthesize all. Some only they will synthesize. So the animals which they can synthesize or prepare amino acids, they are called as non-essential, the non-essential amino acids, which cannot be prepared is essential amino acid. Means out of 20 in human, 9 are essential, means we cannot synthesize those things. 11 are non-essential, means we can synthesize those amino acids. So why the amino acids are he telling essential and non-essential? If the protein has to be made means all the 20 types are required. But if some amino acids we are unable to synthesize, then naturally we have to take from the outside in the diet. To the diet only we have to take. That he has mentioned in the textbook that I am telling. Through the diet which we cannot synthesize the amino acids can be taken. Those are called as essential amino acids. There are 9. And remaining 11 are non-essential, we can synthesize them. So nutrition value means what that amino acid, if it is essential amino acids are not available, some proteins we cannot synthesize, our growth will be, our metabolism will be damaged. That only he want to tell. Now essential amino acids cannot made by the human and non-essential can be made by us. This is he has given in the textbook. So what you learn from the protein? Proteins are the heteropolymers made up of the amino acid, 20 types of the amino acids. And next, whenever we are seeing, they are the linear and they are also called as polypeptides because amino acids in between whatever the bond is the peptide bond. And it is amino acid, whatever the proteins are the heteropolymers because 20 types of the amino acids act as monomers, but different monomers used to participate in the formation of protein. So it is heteropolymer. And according to whenever we are seeing its nutritional value, whenever we are taking the protein into the body, some of the it will be converted into the amino acids. From those amino acids, our body can synthesize whatever needed proteins. In that one essential amino acids which we cannot synthesize in our body, those amino acids should get from the diet only. They are the 9, it is out of, but 9 it is. And 11 are the non-essential. Non-essential can be synthesized by the body. That is the thing he has given. And next, these proteins are very, very important for the functional. Whereas further some protein and their functions. On this one, based on this one, 3 questions given. Whatever the tables he has given in the biomolecules, all are important. From there only he is picking all the bits, whatever the neat points are there. In this one, all these he has given as the matching in the 2024. And next, only about the glut 4 he has given in the 19. And next, like that they are giving. So you have to read these things. Now we will see these functions. Collagen protein is, it glues the cells in the tissue. We are having the unicellular or the multicellular used to attach one to another. But human also it is a multicellular. So how the cells will be glues means it will be at a intercellular ground substance, intercellular cement also we can talk. Now I want to tell you one point. This is in the animals. In the plants, what is that? Middle lamellae. Go and see there. Like that you have to prepare. That is made up of the calcium pectate he has given in the cellulose, in the cell, whatever the cell wall preparation he has given. Next, trypsin. It is an enzyme. All the enzymes except ribozyme are the proteins only. So enzyme, it is trypsin. And insulin, sugar patient used to use, no, that is the hormone. It is also protein. And next, antibodies are also nothing proteins. Remember all the things. Antibodies are nothing protein. And receptor means whatever the sense organ, sensory receptor, smell, taste, and hormone, etc. Means all these sensory organs used to work based on the proteins. That is the thing. And GLUT4 enables the glucose transport into the cell. What is this? Here one more point is there. It is unable to, means it will work, GLUT4 will form a channel to entry of glucose inside. The proteins which is to facilitate the entry of the molecules, already studied in the cell membrane. That is the GLUT4 is the protein which is to make the channel for the entry of glucose into the cell. These are the things, at least biomolecules, what are the tables are there. Every 10 days, 15 days, monthly, twice, thrice we have to read once. Means just, it will be, when you are practicing, it will become very easy. Now, one more thing he has given as proteins. What I told, proteins are the basic bodybuilding units of the cell. Such type of proteins whenever we are seeing in the animal, means protein is important for the living organism. And which protein is abundant, means more very high amount it is to present in the animals, is that is called as collagen protein. There we saw, no, collagen protein is the, it is what he has given, intracellular ground substance, intracellular cement, like this it is to attach. That protein is highest amount in the animals. But in the plants, if you see, the RuBis CO enzyme in the photosynthesis it came. Ribulose bisphosphate carboxylase oxygenase enzyme. Enzyme is already told, enzyme is nothing, it is a protein. That is in that, what is RuBis CO abundant on the total biosphere. Means all the living organisms, if you count which urine is the highest amount is to present, means RuBis CO, because plants are the producers. Producers amount will be more. And plants are the photosynthesis they have to do. Photosynthesis they have to do. Carbon fixation is one of the point. In the carbon fixation, RuBis CO used to take mere part. So the amount of the RuBis CO enzyme, that is the protein, is the totally biosphere it is abundant. These two are important for NEET. In the animals, he has once the animals collagen protein. And RuBis CO, it is in the carboxylation process in the photosynthesis. So up to here we completed proteins. Now we'll see the polysaccharides. So polysaccharides, how the polysaccharide made up of the sugars. Polysaccharides are the made up of the sugar. It is a polysaccharide is a polymer made up of the monomer, that is the monomers of sugars. Are how they are the thread like, means if you take the cotton fiber, you can see thigh also, thin fiber, that is nothing, cellulose, made up of the cellulose, that is the, so thread like structure, the what they are, polysaccharides are thread like structures contain different monomers or same type of the monomers. Different monomers are present, then it is the heteropolymer. Same type of the monomers are present, homopolymer. So as a monomers. So monomers here, and one more thing, remember, repeatedly I am telling, homopolymer, heteropolymer can be seen only in the polysaccharides. Proteins cannot sense, proteins are not having any homopolymers, and nucleic acids are not having the homopolymers, heteropolymers. But here, homopolymer, heteropolymer are present. Now, cellulose is a homopolymer made up of the units, that is, monomer is called as a unit. The monomer are, what are the monomers? Glucose, means glucose, like that. If you want attaching, then it will become cellulose. So next will be the starch, and next one more is the glycogen. Starch and glycogen, this is important need, are also made up of the glucose. Then what is the difference? What is the difference? Glucose and cellulose is made up of monomers of glucose, and starch, glycogen also made up of the glucose. That what is the difference in between the three? Important. Cellulose is the linear. There is no secondary structure. But whenever you see the starch, it is a helical structure. Means the secondary structure can be seen in the starch formation. And branches used to present in the glycogen. Branched structure. Remember all the 3 points. He has given it directly and indirectly in the textbook also. Linear structure is cellulose. Secondary helical structure is starch, and branched structures used to present in the glycogen. And this whatever the starch and glycogen we are telling, starch and glycogen used to store the energy storage. And what the difference? What is the difference? All three are made up of glucose. What is the difference? Only there varies in the structural difference. Already I told structural. One is linear, one is helical, one is branched. That's all. Difference is structural difference. Based on that one, we are giving name. And cellulose will work as the cell wall formation, and glycogen and starch will be used as the storage of energy stores, storage of the energy there. So varies, and polymers made up of the monomers, glucose, and both storage house of the energy. Which one? Starch and glycogen. And next, whenever we are seeing, this is more important. One more he has given in somewhere in the before 20, he has given this one. Inulin is the polymer of the fructose. He has given only sentence, not mentioned anything about it, but I will tell. Fructose means it is to, from the fruits it is to come. Fructose monomers, homopolymer is forming. It is called, he will ask. Inulin is polymer of fructose. Only glucose 3, cellulose, starch, and glycogen. But here, inulin is the polymer of the fructose. Fructose used to form. Fructose is not digested by us mainly. This way out of it is. And out of syllabus, I am telling for your information, for your more attention. Fructose will be used for the kidney functioning test. That is not necessary to you. And one more thing, the starch and glycogen can or glucose, cellulose, we can in the test by testing them, which one is glucose, which one is the cellulose, which one is starch, which one is the glycogen, we can see, we can decide by the iodine test. Iodine test 8th class, 9th class he has given. When you applied the iodine, if it is turning to the blue color, then it is starch. If it is not turning, that is the cellulose. And if it is turning into the brick red, that is the glycogen. But only he has given only 2 points only. That is, in the secondary structure, helical structure is present in the starch. In the helical structure, in between the helical structure, the iodine molecules will be present, so they will be converted, they will appear as blue color he has given. But in the cellulose, it is linear like this, there is no gap in between them, so there is no chance of the hiding of the iodine particles in that one, so it won't give any color he has given. That he asked in the 2023 one bit in this one, polysaccharides. And inulin in the matching he has given before 20. And inulin is the monomers or the fructose made of the inulin. Once he has given in the olden days, insulin, inulin are. Insulin is a protein, inulin is a polysaccharide. That's all. See, how he will catch such type of things also he will catch. And polysaccharide and the monomers, whenever they are attaching one to this unit like this, it is forming a chain. They used to have attaching will be the one angle, that is the non-reducing side. Reducing and non-reducing ends used to present. Reducing end means it can be able to attach to other molecule, other monomer. Non-reducing end, direct non-reducing end cannot be, this is reducing and non-reducing. It will extend like this from the right hand side, not left hand side. That is the thing. That is first, these are, for example, first, it is generally the sugars are the cyclic. First carbon and fourth carbon. First carbon is non-reducing, fourth carbon is non-reducing. Right side is reducing and left side is the non-reducing, and non-reducing end will be the fourth carbon and reducing end is the first carbon. That much you remember it. Because first carbon, second carbon, third carbon, fifth carbon, it will come in the DNA detailedly. For that one I am telling, otherwise I won't discuss. Fourth carbon will be used for extension, means it will attach to the other non-reducing reducing end and forms the chain, that is polysaccharide. So reducing and non-reducing end you came to know. Now here you see one more, and next will be the cell walls. Cell wall is made up of by the cellulose, majority. In the cell wall of the plant made up of by the cellulose. So next what it is, why we are telling that much? The cellulose will be useful for the preparation of the paper. So for preparation of the paper, which polysaccharide is useful? If you can ask, indirectly you can ask the question. Polysaccharide is what? And or you can complicately also you can. Paper using polysaccharide is homopolymer or heteropolymer. What is that? That is homopolymer, because cellulose is the glucose. He is not asking like these questions. If we start asking means Even intelligent students also somewhere he will lose marks. But I am expecting such type of paper because already what happened, in the recent 26 2026 exam cancelled. Very useless paper he has given, but they want to tight the papers. So in this way also this is why he has given that cell walls made up of the cellulose, and the cellulose will be useful for the preparation of the paper. How they will prepare? First they will separate the cellulose in the form of paper pulp. Paper pulp will be prepared from the cellulose which is present in the plants. Generally bamboos they prefer. And then this paper pulp will be used for the preparation of the paper. So first prepared paper pulp, that both the words he has given. First prepared the paper pulp, then it will become paper. And both will be done by the cellulose. Cellulose is the polysaccharide and it is a homopolymer. And it won't convert into the color because it is a chain like it won't catch the iodine. It cannot hide the iodine in that. All these are about the cellulose only. Next, one more thing is, up to now we are seeing the simple sugars. Means generally they used to monomers, they act as. Some sugars can be formed with some complex sugars. For example, he has given that is sugar attached to the amino acid, amino sugar, that is called as a amino sugar, which is called as complex sugar. So complex polysaccharides are amino sugars like glucosamine and next N-acetyl galactosamine. He has given the two names. You have to remember the thing. Glucosamine, N-acetyl galactosamine, both are the complex sugars. So what? Now the complex sugars used to form amino sugar, the exoskeleton of the insects, cockroach exoskeleton plates used to present. What you read? That is the chitin. And the same chitin cell wall used to present in the fungi also. Remember it. So chitinous exoskeleton of arthropods is chitin. But he has not given chitin contains that is the complex sugar, amino sugar. But just for information I am telling you, this is the N-acetyl glucosamine. N-acetyl glucosamine, not galactosamine, N-acetyl glucosamine used to present in the chitin, which is to present in the exoskeleton of the arthropods and the fungi also. Remember the point. Not given there, but he has given clearly in the biological classification. Chitinous cell wall used to present in the fungus he has given. That's all. Next, whenever we are seeing, one more important point he has given, that is all the complex sugars are formed with the means amino sugar plus amino acid. Whenever they formed one unit, the units will continue like that as a homopolymer. That only it is most important point. One of the complex polysaccharides are mostly homopolymers. This is important. Means some other thing, this thing, generally it won't form. Complex sugars used to form the homopolymers. That is also important one. Now we'll see about the nucleic acids. Nucleic acids are most important for the genetical material it is. But nucleic acid is the big macromolecule and it is having that lot of molecular weight is about 10,000 daltons. That already we studied in the last time. So nucleic acids are polynucleotides. It is a polymer made up of the nucleotides. So it is polynucleotide. Polysaccharide we told. Here it is polynucleotide. Nucleotide is the monomer of the which is forming the nucleic acid. So the nucleotides, so nucleotides are the three types of three components used to present in one nucleotide. What are they? Heterocyclic nitrogen base, one is, and monosaccharide ribose sugar, and phosphoric acid or phosphate. This I am not sure here, but textbook also not given the diagram, but these things will come detailedly in the molecular basis of inheritance, DNA structure, RNA structure, there it will come again. So just outlines only, just you see here. So heterocyclic nitrogen base. Heterocyclic nitrogen compounds are present after the lipids he has given. That is the nucleoside and nucleotide. He has given all the things I discussed in the previous video. And heterocyclic means it is a cyclic like benzene, but not all the carbons, different used to present. That is a heterocyclic. Nitrogen bases used to present. And monosaccharides like ribose sugar. Ribose sugar is important for the formation of the nucleic acid, nucleotides, and phosphoric acid and phosphate. Either phosphoric acid may be present or the phosphate it is may be present, because both are easily can convert one to another, and students used to get the confusion. Some places in the DNA used to phosphoric acid, some places used to say phosphate. That is easily convertible. Now you see nitrogen bases, whenever we are seeing heterocyclic nitrogen base. Heterocyclic nitrogen bases are the 5 types are present in the 2 groups. 5 types. Nitrogen bases are how many? 5 types. How many groups are there? 2 groups. First group is, that is the purines, and second group is the pyrimidines. Purines and pyrimidines are the groups which are present in the nitrogen bases. Nitrogen bases not a number is the 5. Out of this one, purines are the 2, pyrimidines are the 3. So adenine and guanine are the purines. And already there also I told you, adenine and guanine, one diagram he has given there, double ring it is to have. The purines is to have the double ring. And cytosine, thymine and uracil, they are the pyrimidines, they are the single ring. They are the single ring. That much you remember it. And next will be the sugar ribose sugar. Based on the ribose sugar only, we used to call the nucleic acids the RNA or DNA. This is also neat point, because ribonucleic acid and deoxyribonucleic acid, DNA, RNA ribonucleic acid, RNA DNA deoxyribonucleic acid. What is this? Ribonucleic acid means ribose sugar. Deoxyribose means on the second position, whatever the hydroxyl group is there one oxygen will be missing. So it will be dehydroxy. Deoxyribonucleic acid. We used to call it as deoxyribonucleic acid. And the second position hydroxyl group, that we will see detailedly in the molecular basis of inheritance. This point. So DNA deoxyribonucleic acid, and RNA means ribonucleic acid. Those are the things he has given, and these are the things about all the things. And this now you see how many points are there for the statements matching. Different type means he has given, but generally he is not touching means chances are there he will touch, because it is not coming from there, students will neglect it. But whenever he is touching, again all the people will run behind that only. It is not like that. If he is not touching, then we have to more concentrate on that one. Such like that only you have to prepare. And if you like this video, please give the like mark and subscribe the channel. Thank you.